لللينير كروموزومز فيه مشكلة. لو تسكروا الباب وراك صوت عالي. يا ريت. سكر الباب. اوكي نعم. ايش؟ براد؟ براد. اوكي سكر. طيب في عندنا مشكلة بالريبليكيشن لللينير كروموسومز اللي هي باللاجينج ستراند. لو عندي انا leading strand راح تضلها مكملة 5 برايم لا توصل للأخير لحد ما ايش بصير بالبوليمريز بوقف انتهى خلص كل شيء. المشكلة باللاجينج ستراند انه بنعمل اوكازايك فراجمنتس 1 2 3 لما وصلنا للأخير there isn't enough space للDNA primase انه يجي يحط برايمر. There's no space. So what? Okay. So that's a problem. The problem here. إنه هذا ما كمل. هاي السيل آخر شيء ما كمل الدينا replication. فلما يصير في another round of replication اللي راح يصير إنه هاي الستراند اللي هي ما كملت it's short. فالتمبليت تبعها أو the other strand will be short as well. معناته with every round of replication صلحوا with every round of replication ايش راح يصير؟ The chromosome will be getting shorter and shorter. هلا if you can imagine احنا لما يكون عندنا embryonic cells لما يكون عندنا embryonic cells لما يكون عندنا fetal cells وحتى لما يكون عندنا cells تبعتنا our own cells and we have a significant number of DNA cell divisions and DNA replication معناته ايش راح يكون؟ That's a huge problem لأنه the DNA will be getting shorter and shorter. The problem is احنا بهاي region هذا راح احنا بهاي region at the end of the chromosomes we call it telomeres. تيلو يعني ends نهايات. في وقفة معينة مثلا. اوكي. اسمها telomeres. التيلوميرز الفانكشن تبعها للدي is so significant لأنه what they do إنه they stabilize chromosomes. So when they get shorter and shorter, chromosomes and they're lost eventually, chromosomes become unstable. بمعنى إنه chromosomes would break up into يعني it would be easier for chromosomes to break up. It would be easier for chromosomes to be mutated. So telomeres are really important. So there is a solution. The solution is a hero known as telomerase. Okay? This enzyme is really an amazing enzyme. What it does, it is a reverse transcriptase. It uses a template, and this template is an RNA template. Okay? That's one. The other thing, this template is part of that enzyme itself. Just like saying glycoproteins. Lipoproteins. اللي هم proteins with sugars or proteins associated with lipids associated with proteins. We have what is known as ribonucleoproteins. Proteins with a non-protein part, which is RNA, in case of the telomerase. Okay? So this template, which is part of the enzyme itself, the RNA molecule, it is used, the telomerase, what it does, it hybridizes or it binds to the telomeres, and this template is used to elongate the DNA template itself. Okay? So this gets longer and longer and longer, giving or creating enough space for the primase to come in and add a primer. ماشي? So what happens, this gets longer and longer, and the primase can come in, it can add a primer and extend the final Okazaki fragment. And I showed you this animation before. How it gets longer. I hope you watch this at home. Please do. Okay? Hello. Notice something. Since we're using the same template, we are repeating the same sequence. And yes, telomeres are made of repetitive DNA sequences, which are GGG-TTA. So you will have this sequence repeated like thousands of times in the chromosome itself. Now, in telomeres. And remember, telomeres are what we call non-coding. Non-coding, yani they do not code for proteins. They are not used as genes to synthesize or to produce proteins. Okay? Now, yes, we're doing polymerase. So it is a reverse transcriptase. Yani it functions as a polymerase, creating DNA, except that it uses RNA as a template. عشان يسمينها reverse transcriptase. Okay, I don't know how to make this faster. Like I put this note right here. This animation is wonderful. It's nice. Like it contains some errors. If you can find these errors, that would be great. يعني في أشياء اختلافات بين what I've mentioned and what exists in this animation. Like in the idea, I hope that you will at least get the idea behind it. So try to be careful. Hello. I have bad news for all. Eventually, all of us will die. That's a fact. And we die because we age. And when we age and we die, it's because of something like organ failure. Organs are not functioning well anymore. Layers, the cells are dying. Why is it that cells are dying? Because chromosomes are not stable. Why is it that chromosomes are not stable? Because telomerase, the enzyme itself, the activity and the amount of enzymes in aging cells, or as we get older, both of them, the level and the activity of the enzyme, get reduced. So it cannot handle or it cannot elongate chromosomes anymore as cells are replicating their DNA and as cells are dividing. And since chromosomes are not stable, cells would die. فعلشان هيك سموا التيلوميريسز انه هم هدول اكسير الحياه، انه هدول راح يخلونا نعيش لفترات اطول. And they found out that cancer cells, since they replicate their DNA continuously, since they are dividing cells, highly dividing cells, they found out that the level of telomerase and the activity of telomerase are really high in cancer cells. But they thought of treating cancer cells by inhibiting telomerases. Yeah, it's all under work, under research. Okay, now. حتى the stem cells, just like cancer cells, they have high level of telomerases. Now. بكل telomerase? Yes. Any replicating cell must have telomerase. Definitely. Okay. ليش بنموت؟ لا عنا. Of course. Okay. Let me say that again. So all cells have telomerases. All cells that replicate their DNA have telomerases. Okay? In younger individuals, in embryos, fetuses, and in children, the level and activity of telomerases are really high. فعشان هيك chromosomes are preserved and maintained. As we age, the activity and expression level of telomerases are reduced. So as cells are replicating, chromosomes are getting shorter, and cells eventually die. Okay? The activity and the level, the amount, الاكسبرشن تبع الجين اللي بيعمل التيلوميريز. نعم.
لو صح المفروض انه احنا بس نولد يكون عندنا سوماتيك سيلز يعني طول معين انه ما يزيد بس ريبليكيشن يكسر بس صحيح المفروض ما يزيد بالضبط ما يكون
المفروض ما يكون في تيلوميريز.
السوماتيك سيلز
بالسوماتيك سيلز ليش؟
لانه له طول معين.
طيب
ريبليكيشن يكسر
ما هو بيحافظ على طول التيلوميريز.
لا هو لازم يكسر طول لازم يكسر لحد ما نخليه يفوت مش انه اذا جسر رجع يفوت. يعني الستيم سيلز المفروض يكون تيلومير عشان كل انسان ترجع
حتى السوماتيك سيلز.
لا السوماتيك سيلز المفروض
مين قال لك انه هلا مش لا حتى السوماتيك سيلز يعني مثلا السوماتيك سيلز بتعمل ديفيجن بس
تكسر
بس اذا بتقصر واحنا صغار مصيبه لانه كل السوماتيك سيلز رح تموت. هلا مش كل السيل رينيوال اللي بجسمنا مش كله من الستم سيلز. مو كله من الستم سيلز، في سوماتيك سيلز بتعمل ديفيجن.
بتعمل ديفيجن طبيعي صح.
طيب
بس بيصير طولها.
ايفنشولي يعني واحنا صغار لا ما راح الكروموسوم ما راح يقصر طوله واحنا صغار في السوماتيك سيلز لان التيلوميريز راح تكون الاكتيفيتي تبعتهم عاليه والاكسبرشن عالي. لكن لما نكبر بيقل الاكتيفيتي تبعتهم حتى السوماتيك سيلز. اوكي؟ طيب نعم. اوكي سؤالها يا اولاد الله يسعدكم هدوء. سؤالها انه هل الاكتيفيتي تبع التيلوميريز بتختلف من شخص الى اخر؟ I think that's my opinion. I think انه في individual variations. لكن يعني مش يعني واحنا صغار مش كثير في individual variation. Maybe as we get older الفاريشن راح يصير اكثر بين الاشخاص. فعشان هيك يمكن بتلاقي واحد بموت عمره 90 سنه وواحد بموت عمره 60 سنه. Is it related للتيلوميريز؟ Could be possible. لكن I think الفاكتور تبع اللايف ستايل وال يعني playing sports and all of that plays a more important role than ممكن انه ايش مره ثانيه؟
يعني اول ما يشوفوا الاكتيفيتي تبع التيلوميريز يشوفوا يعني يتوقعوا كم بيعيش.
اوكي let me promote myself شوي هيك اتفشخر. I have a channel on YouTube في فيديوز على نيوز اخبار بالنيوز ويعني بتصلوا معي وبطلع نيوز and so on. في واحدة من الاخبار خبر طلعوني عليه I think BBC I believe مش متذكر بس كان بيحكي على انه scientists are trying to predict how long an individual would live based on the genetic profile تبعه. Okay? So watch this. Yes, watch this. I think it might answer your question. فممكن يعملوا prediction هذا الشخص لأي فترة راح يعيش يعني كم سنة based on the genetic profile. Okay. You remember Dolly, huh? You've heard about it. هاي النعجة اللي عملوا لها cloning. و 2 weeks ago I think عملوا cloning لmonkeys. So, human cloning possible? Do you think we should do human cloning? I don't know. Why not? I don't know. I just keep asking myself this question. Why not? Think about it. Okay, that's up to you. All right. Dolly. هلا أنا بعرفش كثير بالغنم. بس let's assume إنه الغنم بيعيش 10 سنوات. Dolly was born day 1. She was day one old, one day old. It lived 5 years only. Instead of 10 years, it lived 5 years only. And أمراض الشيخوخة، تجاعيد الوجه، العظام المهتكة and so on بينت عليها early on. ليش؟ لأنه the DNA that they used was actually a 5 years old DNA. So it was already shorter than normal. So Dolly didn't survive for a longer time, and diseases and certain conditions appeared on Dolly early on, of old, symptoms of old age. Okay, so let's go on. بما إنه حكينا على chromosomes and stability of chromosomes and mutations, let's go on with mutations. In general, we can classify mutations into two types. We have macro mutations and we have micro mutations. Macro mutations are large mutations that can appear at the level of chromosomes. So we have, for example, an extra chromosome. We have one chromosome missing. We have an arm of chromosome that is, let's say, duplicated and so on. So لما نطلع على chromosomes under the light microscope, we can see that there's something wrong with this chromosome. That is genetics and genetic mutations. I'm not going to talk about these. I'm concerned more with micro mutations that occur at the level of DNA. Okay? Things that we have to use more advanced instruments than light microscopy to observe. There are different types of such mutations. We have the most common is what we call single point mutations or single base mutations. So basically, it's a mutation in one base. Of course, we cannot see that under the light microscope. And single point mutations, there are different types of such mutations. We have what is known as silent mutations. A silent mutation is a mutation that does not cause an alteration in the protein sequence, in the amino acid sequence of a protein. Okay, so it stays the same amino acid. There's no effect on the protein itself. How about a silent mutation? Is there a mutation? It is a mutation. There's a change in the DNA, and it doesn't have a major effect at the protein level. This is silent. Now we have what is known as a missense mutation. Sense yani logic. Missense is basically a change in one base resulting in change in the amino acid. So like sickle cell anemia, instead of glutamic acid, we have valine. Just a change in just one base. Then we have what is known as a nonsense mutation. Nonsense mutation هو basically termination of protein synthesis. So you have a truncated protein. You have a shorter protein. The severity of this mutation depends on where it exists. Yani if it exists early on in the gene or the protein sequence, that means that you will have a defective gene, a defective protein. Like if it exists at the end of a protein, it may not have a major effect. So severity depends on where the mutation takes place. And then we have what is known as harasmon nonsense. And then we have frame shift mutations. You probably already know that. Like we have every 3 nucleotides can code for an amino acid. These are called codon. So every one codon, 3 nucleotides, gives us 1 amino acid. You can imagine that every codon is placed in a frame. برواز. And it's a smiling codon. فعندنا frame, and we have another codon in a frame, and we have a third codon in a frame. كويس؟ Let's say that the first one is 1, 2, 3, second frame, 4, 5, 6, third frame, 7, 8, 9, and so on. Let's say that we insert a base between number 2 and 3. So in the first frame, we will have 1, 2, A, base A, 1, 2, A. Second frame, instead of having 4, 5, 6, we will have 3, 4, 5. Okay? But then we will have 6, 7, 8. So we are changing the contents of the bases within a frame. So what we're doing is that we're shifting the frame. Okay? And this is known as frame shift mutations. As a result, such mutation causes a major change at the level of the protein. Then we're creating a whole new sequence of amino acids. Again, the severity of such mutation depends on where it exists. If it exists early on, everything that comes afterwards is different. If it occurs at the end of a gene or a protein, it could be minimal. Then we have other types of mutations that occur at the level of DNA, translocations, movement of a small part of DNA to another region. We have deletions, 3 base pair deletion, insertions, duplications, and inversions. Different types. Now, ما هي المحاضرة؟ كل المحاضرة على الميوتيشنز. لا هذا استنى على شوي. هديه من روعك. So what are the causes of mutations? We have induced mutations and we have spontaneous mutations. Spontaneous mutations, they happen without any external factor. They just happen within the cell. Normal. It's a normal process. It's abnormal. It can be pathological, like it happens. And then we have induced mutations. That is, we have an external factor outside the cell, outside our body, like sunlight, for example. Okay, it can cause mutations. But نيجي نحكي عن spontaneous mutations. We have such mutations can be related to errors in DNA replication. Meaning that the polymerase itself makes a mistake while reading or while synthesizing the DNA. And one reason for errors in DNA replication is the existence of tau tumors. Tau tumors is like isomers of nucleotides, or isomers of bases. So you can have thymine and you have its isomer or its tau tumor that is different, a little different. صار في change بالstructure of thymine itself. And a part of it, the DNA polymerase doesn't look at A in the template and says to itself, where's T, where's T, where's T? It doesn't look at T. It looks at something that can fit into the active site, 1, 2, that can form 2 hydrogen bonds with this thing that exists in the template, that is A. So it can take something that forms 2 hydrogen bonds, fits into the active site, and it forms 2 hydrogen bonds with the A. If it sees G, it looks for something that can form 3 hydrogen bonds, like C, and it fits into the active site. So hadolic tau tumors, there's interconversion of the bases themselves, whereby instead of GC, AT base pairing, you can have AZ, and you can have GT. Why? Because the tau tumor of T can form 3 hydrogen bonds. The tau tumor of a normal C can form 2 hydrogen bonds. So C now fits with A, and T fits with G, and so on. فهذه tau tumors, the concept of tau tumors. We have frame shift mutations that DNA polymerases can create within the DNA. Frame shift mutations, هذول disastrous mutations. These such mutations, as well as deletions and duplications, they exist mainly in regions of DNA that contain repetitive DNA sequences. يعني ايش؟ Let's say specifically نحكي كexample CAG. Certain genes راح نحكي عنهم كمان شوي. Certain genes contain repeats of CAG. So you have within a gene, you have CAG, 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 CAG. All right? هلا what the DNA polymerase does إنه CAG, 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 CAG. Let's imagine it this way. The DNA polymerase gets confused. How many CAGs have I added? I don't know. 10? Let me add more. Oh, let me stop. If you have C, 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 well, again, same thing. How many C's? I don't know. Let's add more, let's add less. So polymerases can get confused, and especially in sequences that are repeated, that they have repeated sequences, or repeated nucleotides. Okay? So, creating frame shift mutations. يعني مثلا عندنا 4 C's, C, 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 C. It can add 1. That can cause a frame shift mutation immediately. And there are certain diseases and conditions that are related to mistakes within repeats. Just like a mitochondrial condition known as Kearns-Sayre syndrome, we have results from deletion in repeats, or we have increase in number of repeats, such as in these conditions. Fragile X syndrome. You will study Fragile X syndrome in genetics. Basically, it is a gene, there's a gene FMR1 that exists on the X chromosome and has this repeat, CGG. And it exists at the end of the X chromosome. Now, the number of repeats with the Fragile X syndrome can increase to large number. So normal individuals have, let's say, less than 50 of such repeats, the CGG. Like affected individuals have like 500 of these repeats. Huge. That causes destabilization of the X chromosome, and the X chromosome gets broken. Literally. عشان هيك بسموه fragile X syndrome, يعني قابل للكسر. We have other conditions related like myotonic dystrophy and Kennedy disease, Huntington disease. This is really a fatal disease, Huntington disease, all related to such repeats. And CAG repeats with androgen receptor. This is something that a gene that we're working on in our laboratory. Then we have spontaneous lesions. يبقى كل اللي حكينا عنه قبل هيك كان errors related to DNA replication. هلا برضه عندنا spontaneous mutations إنه ممكن يكون في lesions that occur in the DNA. Lesions, جروح, أخطاء صارت في DNA, like depurination, deamination, or the action of reactive oxygen species. إيش يعني depurination? Depurination, here you have a nucleotide as part of the DNA, and it contains a purine. All of a sudden, it can happen that you have dissociation of this purine. So you'll have a nucleotide that has a sugar and a phosphate, but it does not have a base. It just happens spontaneously. So the DNA polymerase comes in, it reads the DNA. Hell, when it reaches this depurinated nucleotide, there's no face, doesn't know what it's reading. It's decision time. It's decision time, and cells must decide. What should I do in this case? Should I kill myself? Well, should I continue with the DNA synthesis? If the cell can go on with DNA synthesis, they will do it. يعني لو اجى واحد ضربك كف تنتحر على طول؟ No. You will try to handle that situation so that you can survive, so that you can fix the problem later on. And cells Just like us, they have souls. They love to live. They don't kill themselves easily. So, فالpolymerase بحكي depurinated. I don't know. What should I do? What should I do? What does it do you think? It adds any nucleotide, anything, so that it can go on. I see? And it goes on. There's 25% chance the added nucleotide is the right one. See? And we have 4 options. So وممكن تكون الريجن اللي صار فيها depurination not so important. And it doesn't affect the individual at all. So what? حطينا A ولا G ولا C ولا T, so what? So the whole purpose is for the cell to stay alive. Deamination is also a spontaneous process that occurs where you can have cytosine when it's deaminated, removal of the amino group, forming a ketone. Well, that changes cytosine to uracil. لو كان cytosine methylated, and this is normal in our genome, cytosines can be methylated, and if it gets deaminated, it gets converted to thymine. So instead of having GC, you will have AT. طب والuracil, الuracil can be repaired. يعني بيجي DNA polymerase بحكي whoa uracil, you shouldn't be here. فايش بحكي؟ You know what? You could be a T بس صارت mistake. يلا راح احط A وبعدين بصلحك بعدين بشيلها. اوكي؟ ممكن انه الادرينين يصير له deamination بتحول لها hypoxanthine. Hypoxanthine it can form a nucleotide, and it's not part of the DNA. Like if it exists in DNA and it can be fixed, it will pair with a C. Again, instead of having AT, you will have GC. Okay? So this is all spontaneous. But with induced, there are different mechanisms for induced mutations. Yani we have external factors. Basically, you can add a base analog to DNA, you can alter an existing base, or you can damage the base itself. طب ايش يعني ايش هم البيس analogs? They look like bases, يعني things that you can ingest. وبيستخدموهم in treating cancer. The cancer chemotherapy. We fool cancer cells since they are dividing by giving them base analogs that damage the DNA, that can stop DNA replication عشان هدول proliferating cells die eventually. So you can have a base analog مثل 5-bromouracil. And a 5-bromouracil, it can be placed in front or complementary to A. خلص الأمور حلوة عال. This can be repaired later on. لكن let's say that it's not repaired وصار عندي DNA replication وصار عندي ionization لل 5-bromouracil, and this is again في عندك interconversion ما بين the normal form of 5-bromouracil and the ionized form. The ionized 5-bromouracil pairs with G instead of normal pairing with A. فلما يجي DNA polymerase ويقرأ إنه عنده 5-bromouracil, what is that? وبشوف إنه بقرأ الجي وبعدين أخذ الionized form حط الionized form عند الجي لأنه it doesn't look for a C, it looks for something that can fit the active site and it can form 3 hydrogen bonds. عندنا كمان من induced mutations we have alkylation of a base that already exists in the DNA. فعندنا guanine can be alkylated, addition of a methyl group, and if the DNA polymerase is synthesizing DNA and it sees methyl guanine, it pairs it with thymine instead of cytosine. Again, we have a single point mutation. أو ممكن يكون عندي base damage, the base itself is damaged, أو ممكن يكون عندك creation of what is known as AP sites. AP sites يعني apurinic أو apyrimidinic sites. حكينا على spontaneous lesion اللي هي depurination. That's normal. لما يكون عندي depurination, this is known as AP site, apurinic site. ما فيهوش purine. أو ممكن يكون عندي strand breaks, يعني break بالphosphodiester bond, and usually the sunlight, the UV light, can damage the phosphodiester bonds. finally على ال induced mutations في عندنا chemicals that exist in nature اسمهم intercalating agents that look flat and they insert themselves between bases. So لما يجي ال DNA polymerase and it's reading the DNA بتشوف في intercalating agents, they can either add a base or they can delete a base, creating frame shift mutations. وهدول الأشياء they exist in nature as well as in laboratories. طيب هذول different types of mutations. هل بالفود اندستري بحطوا كثير preservatives وبحطوا كثير اشياء او صار عندنا صناعات جديده وبدهم يعرفوا هذا الشيء is it mutagenic or not؟ شو يعني mutagenic؟ يعني does it cause mutations or not؟ هلا the thing is انه هدول mutagenic compounds they can be carcinogenic يعني they can cause cancer لانه all carcinogens are mutagens but not necessarily the opposite, not all mutagens carcinogens. فبدهم يعرفوا هذا الشيء is it الكيميكال الجديد هذا is it mutagenic or not؟ طيب هلا ركز معي بليز لانه هذا حكي كثير مهم عشان تصحصح. اجى واحد اسمه بروس ايمز. بروس ايمز هذا اخترع فكره او اخترع طريقه حلوه كثير. هلا انا ما بدي اشوف ميوتاجن I don't want to test it on humans طبعا. I don't want to test it on animals لانه it takes a long time عشان نعرف حيصير عندهم كانسر ولا لا. So let's test it on bacteria. اسهل شيء نعرف هذا الكيميكال mutagenic or not mutagenic. ليش عمل هذا بروس ايمز؟ really smart way. اخذ بكتيريا سالمونيلا وعمل فيها 3 genetic changes. اوكي؟ واحد في polysaccharide coating حوالين البكتيريا هذه شال الجين so that any chemical added to this bacteria it can enter bacterial cells easily. حلو. هاي واحد. الشغله الثانيه اللي عمله انه خرب repair system، خرب DNA repair system. فأي damage أي mutation بيصير بالDNA it is not repaired. It persists. كويس؟ اثنين معناته راح يكون عندي high level of ايش؟ spontaneous mutations وبرضه induced mutations. 3 وهذه الشغله المهمه كله مهم عشان انه this bacteria can synthesize their own histidine. But what he did, he damaged that gene. معناته in order for these bacterial cells to survive, histidine must be added. Okay? هذا شغله مهمة. And his test is based on something known as a reversion mutation. الidea هي هدول mutated bacterial cells cannot survive in the absence of histidine. طب افرضوا انه صار في mutation بالجين اللي بيعمل هيستidine اللي بيعمل الانزيم اللي بيصنع الهيستidine بحيث انه هذا الميوتيشن رجع البكتيريال سيلز صارت تقدر انها تعمل هيستidine. فسماه الميوتيشن ريفرجن ميوتيشن. اعيد اضرب حالك كفين صحصح معي. So what he did انه هاي البكتيريال cells cannot survive unless we add histidine. لكن افرض بما انه ما عندنا DNA repair system it's damaged حيكون عندي high level of spontaneous mutation. افرض انه صار في mutation بمحل الميوتيشن رجعه خلاه صح. اوكي؟ هذه الايديا بيعني. فحكى ايش؟ حاخذ هاي البكتيريا سيل راح احط عليها الكيميكال. If this chemical is mutagenic معناته it will create random mutations, possibly a mutation in the mutated site, in the same site that is damaged. So it brings it back and it makes it normal again. So now these bacterial cells can survive. يبقى the idea انه بناخذ البكتيريال cells لو زاد عدد الكولونيز تبعت البكتيريا معناته انه صار عندي mutation. معناته انه هذا mutagenic. Got it? طيب باخذ البكتيريال cell. I grow it without anything, no chemical whatsoever. راح يكون عندي high level of spontaneous mutations. It could be انه الميوتيشن راح يصير محل نفس الميوتيشن اللي هو عمله. It makes it normal. فراح يصير في عندي انا bacterial colonies لانه صار عندي spontaneous mutation. لو حطيت انا chemical وكان هذا الكيميكال mutagenic راح يصير عندي two types of mutations. راح يزيدوا عدد الميوتيشنز السبونتينيوس زائد الاندوسد mutations. Got it? قولوا اه ولا لا؟ Right, place. So the idea again, ناخذ البكتيريال cells without any million or let's say million bacterial cells without any chemical two colonies لأنه هذول they are a product of spontaneous mutations. لو حطيت الكيميكال وزادوا عدد الكولونيز معناته we say this chemical is mutagenic. Yes? نعم.
يكون كان بس ما عنده ميوتيشن في نفس الوقت الموقع.
It's all a matter of probability. صح؟ Just like you're saying. It's saying انه افرض انه عمل mutations بس ما عمل mutation بهذا المحل. It's all a matter of probability. احنا بنحكي statistics. ف if it creates mutations معناته there's high probability that it will cause mutation in the same place that is mutated. ماشي؟ طب how can we say إنه هذا الكيميكال is mutagenic or not؟ يعني إيش النمبر؟ النمبر هو and it's all arbitrary بحكي لك إنه if the number of colonies doubles اللي هو البكتيريا مع الكيميكال versus البكتيريا بدون الكيميكال if the number of colonies doubles معناته it is mutagenic. Okay? نطلع على هاي examples. Right here. Bacteria with water. 10 colonies. ماشي؟ هذه ايش؟ result of what؟ ايش؟ ايوه احكوا بسرعة please. Spontaneous mutations. حلو. Motor oil 50. Is it mutagenic؟ يبقى احنا condition if the number doubles. Mutagenic? Yes. صار 5 times more. And yes, motor oil is mutagenic. But yes, alcohol 43 mutagenic? Mutagenic. And it is mutagenic. It's actually the 5th cause of cancer worldwide. Alcohol. Smoking is number 2. Number 2, I think. Drug X, is it mutagenic? No, it is not. And the number is not double. Ships of 5 rows, definitely mutagenic. So only it is mutagenic with all of these preservatives that they add. I don't know what they add. لكن عندي factor ثاني that we have to take into consideration اللي هو liver enzymes. What do liver enzymes do? They detoxify chemicals. So possibly إنه هذا الchemical is mutagenic. لكن once we ingest it, liver enzymes can handle it, making it not mutagenic anymore. Or the opposite. يعني you can have something that is not mutagenic and it can be more or it can be mutagenic as a result of liver enzymes. فايش اللي عمله؟ ايش اللي عمله؟ اخذ الرات ليفر كراش بلندر اخذ الجوس وحطه مع الكيميكال وبعدين اخذ البرودكت وحطه على البكتيريا مره ثانيه. يبقى هلا صار في عندي conversion او detoxification او modification لهذا الكيميكال. خلينا نشوف our previous example. That's without liver enzymes. That's with liver enzymes. Water, is water mutagenic? 10 versus 12. No. طب liver enzymes are they mutagenic? No. طيب ماشي؟ طيب motor oil is it mutagenic? Compare this number with 12. Is it double? No. Even it was mutagenic, like liver enzymes have detoxified motor oil, the mutagenic component of motor oil, so it is not mutagenic anymore, so it is safe for you to drink motor oil. Machine? Alright. Alcohol, is it mutagenic? 50 versus 12. Yes, numbers have doubled. Has the mutagenicity changed? No. No. These two numbers are close, not double. معناته liver enzymes have no role, they have no function in detoxification of alcohol. طيب drug X, is it mutagenic? Definitely 35 versus 12. فبنحكي liver enzymes ايش عملوا؟ They have taken this chemical that is not mutagenic and they have made it mutagenic. Yes? And this happens. But chips أبو 5 قروش mutagenic? Definitely. Has mutagenicity changed? Definitely. It has increased. هلا before you leave معلش باخذ سؤالك بعدين. And please do not send me questions by email. ماشي؟ لأنه أول شيء أنا I would love to hear your questions بالlecture نفسها أو ممكن بعد المحاضرة. ثانيا أنا ما بحب كثير أكتب بالإيميلات، يعني my answers will not ما راح تشفي الغليل. My answers will be yes, no, maybe, I don't know, see me. أكثر من هيك أنا ما راح أكتب. So أنا I prefer that you ask me here. آخر شيء بال 2011 I think 2010 في parents عندهم mitochondrial disease، الست عندهم mitochondrial disease وأنتم عارفين إنه الميتوكوندريا بتيجي من الأم. أوكي؟ فعندها mitochondrial disease لأن الميتوكوندريال DNA تبعها is mutated. So فبدهم يخلفوا بس ما بدهم أولادهم يكون عندهم الميتوكوندريال disease أو الكونديشن اللي عندهم. So what they did, that was in 2010, what they did إنه جابوا donor, female donor، أخذوا الإج تبعتها، أخذوا النوكليوس طلعوها رموها، أخذوا النوكليوس تبعت الأم اللي بدها تخلف، طلعوها حطوها بالإج تبعت الدونر. اللي صار فيها النوكليوس تبعت الأم، السيتوبلازم والميتوكوندريا تبعت الدونر، وعملوا IVF الفيترو فيرتلايزيشن وطلع هيلثي بيبي. 2014 الحكومة أو البرلمان البريطاني أقر هاي العملية وحكى إنه it is safe, it can be done, it's ethical، ما عندناش أي مشكلة. I think 2016 جوردانيان كابل راحوا على المكسيك وعملوا العملية وخلفوا فأخذوا مكسيكان ميتوكوندريال DNA مع اوكي وخلفوا. Do you think it's ethical or not?
Yes.
Okay. هون ندخل بالمولكيولر بيولوجي والتكنولوجي اللي عمالها بتغير حياتنا بشكل رهيب كثير. التكنولوجي مو طبيعيه اللي عمالها بتصير والتشينج اللي عمالها بتصير. انا في ناس ممكن يحكوا ميتوكوندريال DNA just mitochondrial DNA ما بيعمل اي شيء. It doesn't code for phenotypes, it just produces energy. That's it. So what? في ناس بحكي لك لا it's unethical لانه it is a DNA. أنا I think إنه it needs further discussion. It could be unethical لأنه we can use mitochondrial DNA for determining paternity. One لكن زي الأخوة بالرضاعة، إخوة شو عرفنا؟ بس الناس بتحكي بقولوا هدول الاثنين إخوة ما بصير يتزوجوا من بعض. So خلينا نحكي إنه هدول إخوة ليش؟ لأنه عندهم نفس الميتوكوندريال DNA. كيف؟ على إنه الحكي إيش ممكن تحكي؟ نحكي عنها بعدين ها؟ يلا الله معك.